ערב טוב אורחים יקרים, ערב טוב זיווה.
ערב טוב טלי.
אנחנו נרגשות מאוד לצאת לדרך עם כנס העשור המקוון של עמותת צעדים קטנים. את יודעת, התרגשתי מאוד לראות את מספר הנרשמים שהולך ועולה מאז שפתחנו את ההרשמה לכנס. התרגשתי מהקהילה הרחבה שיצרנו. ואני מתרגשת שיש לנו אפילו אורחים מחול.
נכון, משפחות, רופאים, מטפלים, אנשי חינוך והוראה, חוקרים ונציגי חברות תרופות, כולם איתנו כאן הערב. וואו, 10 שנים של פעילות, היית מאמינה?
אני מתלבטת אם יותר נכון להגדיר את זה כחוגגים עשור לפעילות או מציינים. מצד אחד, כשרואים את העשייה הגדולה והשינויים שהצלחנו לחולל בארץ ובעולם, זה לגמרי משמח. לראות את ההשפעה שלנו, ההורים, על הטיפול, המחקר ועל חיי היום-יום של המשפחות בארץ ומחוצה לה, זה לגמרי ציון דרך חגיגי.
לגמרי.
הדרך המשותפת שאנחנו צועדים, השינויים שאנחנו מובילים, חולמים ומיישמים, לא מובנים מאליהם. בייחוד כשמבינים מה היה כאן כשהתחלנו. זוכרת? מצד שני, המחלה הזאת לא פשוטה. כל אחד מאיתנו, ההורים, הילדים והבוגרים, מרגיש יום-יום את האתגרים. כל יום מביא איתו התמודדות חדשה, ותקווה לצלוח גם אותה. לכן, יותר מתחבר לי לפעמים לומר שאנחנו בעצם מציינים עצור, ולא חוגגים.
משפחת צעדים קטנים היא משהו שאף אחת מאיתנו, ולא המשפחות, לא יכלה לעצמה להשתייך אליה. לא בחרנו בזה, אבל אנחנו כאן. אנחנו כאן כדי להוביל את השינוי בשביל הילדים שלנו. ואם כבר אנחנו כאן, אז בתוך קהילה חזקה ותומכת. קהילה שלא מבדילה בין דתות ולאומים. קהילה שהיא משפחה.
אנחנו רגע אחד לפני חג חנוכה, ואני חושבת רגע לאחור. על המקום שהייתי בו לפני 11 שנים בדיוק. זה היה חג חנוכה של שנת 2009. יום שמשי, קצת הפוגה מהימים הגשומים באותו שבוע. אני זוכרת אותי, צועדת עם עמית ועידו שלנו, למכון הגנטי בבלינסון. ביקשו שנגיע. חשבתי שזאת בטח תהיה עוד פגישה שיאמרו שלא. הם מצאו כלום ולא יודעים למה ה-CPK של הילד המקסים שלי גבוה. אבל לעמית הייתה תחושה לא טובה הפעם. הוא סגר מראש את היומן, והוא הרגיש שהוא לא יחזור באותו יום למשרד. זה בעצם יום שלא שוכחים אותו לעולם. זה יום שמשנה לך, למשפחה שלך, ולכל הסובבים אותך את החיים. פתאום אני נזכרת שמזל שהשקעתי בלימודי הביולוגיה בבית ספר ובאוניברסיטה, כדי להבין את כל המושגים המדעיים, אבל לא חלמתי לרגע שאצטרך לעבוד יומם וליל כדי ללמוד את התחום. לקרוא דוחות כספיים, פסיכולוגיה ואפילו תורת המשחקים וכלכלה שנדרשים כדי לקדם מהלכים לטיפולים בילדים שלנו, בילד שלי. לא חלמתי לרגע שאגיע לייצג את המשפחות בכנסים בינלאומיים בעולם. בניסיון להביא לכאן כמה שיותר מחקרים, חומר, מידע ומחקרים. לא חלמתי שאעבוד בצמוד לכל כך הרבה רופאים וחוקרים. הצוות המטפל בילדים שלנו, כל אחד מתמחה ועוסק בתחום שלו. אבל לנו, ההורים, יש את היכולת לראות את התמונה הכוללת של החיים, של הילדים שלנו, לכן אנחנו כאן כדי לעזור ולהוביל את הטיפול הנכון ביותר בשבילם. אבל מה שכן חלמתי, ואם לומר את האמת, אני לא מפסיקה לחלום. זה היום שבו נדבר על תרופה. לא רק לחלום, אלא גם לדאוג לזה, להשיג את זה, בדיוק כמו שעשית בשביל עדי וכל שאר המשפחות. לא לפני הרבה שנים השאיפה שלי הסתכמה בעבודה משמעותית כשכירה בארגון הייטק יציב. לא חלמתי שאני אעמוד בראש ארגון התנדבותי למען משפחות וילדים, ממש כיזמית, יחד עם צוות של נשים מקצועיות שנותנות את הנשמה בעבודת צוות מדהימה לארגון, למשפחות העמותה עם השפעה עצומה בארץ ובעולם. היום אני מאמינה בדרך שלנו, מאמינה בעשייה, מאמינה בקהילה של צעדים קטנים שיצרנו ועושה יום-יום למען המשפחות והילדים.
ואני מאמינה בך, ולכן אני משאירה אתכם בידיים טובות עם טלי להנחיית המושב הראשון של הכנס. אותי תפגשו שוב בהנחיית המושב הבא.
עולם הבריאות בשנה האחרונה גועש ורועש בארץ ובעולם. כולם נעים וחיים סביב המגפה שתפסה את העולם לא מוכן, מגפת הקורונה. ברור לנו הצורך והדחיפות של הטיפול במגפה, אולם הזמן לא עוצר מלכת, והחולים בדושן-בקר חייבים להמשיך לקבל מענה. להם יש מחלה פרוגרסיבית שמתקדמת, והיא לא עוצרת פתאום בגלל הקורונה. שיתוף הפעולה בין משרד הבריאות לצעדים קטנים ממשיך לעבוד ולחיות גם בתקופה הזאת. בחודשים האחרונים נפגשנו עם סגן שר הבריאות במטרה לקדם את מרפאת הבוגרים ונושאים נוספים כמו חקיקה, מרפאות למחלות נדירות, וזה במסגרת הפעילות שלנו בקואליציית מחלות נדירות, וכדי לשמור את הנושאים האלה על סדר היום.
לחבריי מעמותת צעדים קטנים, לכבוד הכנס. רציתי לאחל לכם המון הצלחה, אתם עושים עבודה מאוד מאוד חשובה, חלק מאוד משמעותי מכל הקואליציה של המחלות נדירות. ברור לי שהקרב הוא לא קל, הוא ארוך, הוא לא נגמר, זה קרב לשנים רבות. ממני, סגן שר הבריאות יואב קיש, רציתי לחזק אתכם, לומר לכם יישר כוח, ומאחל לכם, ולפחות ממני, לקבל את כל העזרה שצריך, שיהיה לכם המון בהצלחה.
את פט הכרנו בתחילת דרכנו, אחרי קבלת האבחון. גילינו אישה מדהימה, עם כוחות עצומים, עם ידע נרחב, שממשיכה לפעול ולשנות את עולם הדושן למען כלל החולים בעולם, למרות ששכלה את שני בניה בגלל המחלה. חלק מהעבודה של צעדים קטנים ושיתופי הפעולה שלנו בעולם עם ארגונים בינלאומיים שמתעסקים בדושן-בקר, נולדו בזכות העבודה עם פט. היום אנחנו לוקחים חלק פעיל בקבוצת עשייה של מחקר וטיפול, וכמובן, כמקור בלתי נדלה לשיתוף והעברת ידע בין ארגונים. פט תדבר על האני מאמין שלה במקום של המשפחה, בניהול של המחלה ובארגון החיים, ביכולת ובידע של ההורה להעלות את המודעות ולקחת חלק פעיל ומשמעותי, להיות אקטיבי ולהוביל את המשפחה.
Well, thank you to Tally and Little Steps for inviting me here. It's really a pleasure, and I certainly wish we would be in person. The diagnosis of Duchenne muscular dystrophy is a difficult one. You go home and wonder what it is that you can do. How do you best take care of your child? How do you find the best opportunities and treatment for him? And what does your voice mean and how to use it? You become by definition an advocate because you have to inform your family, your other children, your relatives, your neighbors, the school. You become the person who's educating about Duchenne. And in fact, sometimes you see your primary healthcare provider, and that person says something like, "You'll know more about this than me," which doesn't feel very good because you want everyone to know as much as possible about Duchenne muscular dystrophy. But by default, you become that advocate. You become the educator. You become the lead of the healthcare team to discuss what's right, what might be the best intervention for your son, how to care for him across the years that are going to come forward. You want to think about how do you preserve and protect his muscle strength? How do you preserve and protect that heart? We have standards of care that have been published in this disease, and they should be adhered to. And you, with your healthcare provider, should be discussing what that means and how to utilize it. As therapies come on board, you should be able to take advantage of the therapies. Nothing should prevent you from accessing a therapy for your child. Not where you live, not what insurance you have, not what doctor you go to. If you feel as a patient in a family that this is the best way forward, that's what you take advantage of. I think sometimes when we have a diagnosis like Duchenne muscular dystrophy, we think our voice doesn't count. But in fact, the voice of rare disease patients, and specifically the voice of Duchenne muscular dystrophy, has changed the world. We have been involved in the diagnosis of these babies, launched newborn screening programs so we can diagnose them very, very early. We've been working with healthcare providers to talk about what matters to us. We've developed with our community outcome measures by which we measure our son's progress and his progression in his disease. We've been working with companies over time to talk about the clinical protocols, the testing mechanism that they will determine whether this intervention they're developing has value, whether it makes a difference. And then we, in turn, as patients and families, we explore those therapies and decide if they provide value to us. If whatever the requirement is by this therapy, if whatever the benefit is, means something, if it matters to us. And certainly with governments, raising our voice, talking to them about Duchenne muscular dystrophy, a progressive neuromuscular disease that over time takes away function. And finally, the heart. How can we together think about making the most quality lives for our children? How can we work with companies to make sure the trials are useful, that they come and have a result? Yes, whether it works or whether it doesn't. That we work with our governments to make sure that this child has access to those therapies that work. And at the end of the day, that we live full and healthy lives. The only way that this happens is a collective voice. You, as a family, are the voice of your child. And working together with little steps, you gain a bigger voice and a bigger presence, not only in Israel, but across the world, as we work together to globally change the outcome of Duchenne muscular dystrophy for our own children and for every child that will be diagnosed now or in the future. What we want is quality of lives. I think one of the young men in the Netherlands said it best: if we're not here for a long time, let's be here for a good time. Let's be here for the quality of the lives of our children. Let's raise our voices together. Let's have a consistent message, and let's go on and change the world. We can do it together.
התחלנו את הפעילות של צעדים קטנים כעמותת הורים. עשייה של 10 אצבעות של הורים שהחליטו לקחת יוזמה לידיים ולמצוא דרכים לשיפור הטיפול, החיים והמחקר לתרופה. רק 10 שנים עברו מאז, והיום עמותת צעדים קטנים כבר לא כזאת קטנה. תפסנו מקום משמעותי בעשייה מול רשויות בחקיקה. אנחנו מעורבים בעשייה בלתי פוסקת ומשתפים פעולה עם עשרות חברות רפואיות ומכוני מחקר. אנחנו פועלים בצורה מסודרת מול המרפאות, המטפלים, והחשוב ביותר בקשר הרציף מול המשפחות. כל זאת מתאפשר ומתנהל בעזרת צוות של אנשי מקצוע שמוביל את העשייה בעמותה. בואו נדייק, במקרה הזה מובילות. חבורה של נשים שכל אחת מהן יחד ולחוד מרכיבה את הפאזל שנקרא צעדים קטנים.
היום כל משפחה יודעת שיש לה כתובת, שיש לה למי לפנות, שבכל עניין ועניין הם מרימים את הטלפון ואנחנו בעצם ניתן את המענה.
חשוב לנו בעמותה לשפר את איכות החיים של המשפחות שאנחנו מלווים אותן. אנחנו עושים את זה במגוון רחב של מענים, אם זה טיפול רגשי למשפחות בשלבים מאתגרים שהם מתמודדים איתן, אם זה קורסים וחוגים מקצועיים לילדים ובני נוער כדי להעשיר את עולמם, ואם זה הכשרה מקצועית לבוגרים כדי לשפר את תחושת המסוגלות שלהם ולהכין אותם לחיים עצמאיים.
אנחנו נותנים מענה למשפחות בהיבט המחקרי והטיפולי. המענה מותאם בראש ובראשונה לצורך של המשפחה. למשל, כשמגיע מחקר או תרופה חדשה לארץ, אנחנו מעדכנים את המשפחות הרלוונטיות שיכולות להתאים. המענה דינמי, הוא מותאם גם לרוח התקופה. עכשיו בקורונה יצרנו דרך לקבל טיפול פיזיותרפי מרחוק.
אנחנו עובדים בשיתוף פעולה עם רופאים וחוקרים מארץ ומחול, הפכנו למרכז ידע מקצועי עבור משפחות והצוות המטפל, ואנחנו חלק משמעותי מהשיח העולמי בקידום הטיפול בדושן-בקר.
זה נהדר שיש כל כך הרבה פרויקטים בסיוע למשפחות, בחקיקה, במחקר. אם פעם היינו מממנים את הפרויקטים במחירה של נרות ומשלוחי מנות, היום העשייה הרבה יותר רחבה ומשמעותית וצריכה תמיכה הרבה יותר גדולה.
בתחילת הדרך רצינו שידעו מה זה דושן-בקר ולסייע למשפחות. היום אנחנו יודעים שבכל תחום שניגע יש עוד המון מה לעשות. לשנות תקנות בתחום הניידות, להנגיש מעלית בבית ספר למרות שיש חקיקה, לקדם מרפאת בוגרים כדי שיהיה רצף טיפולי ולהנגיש תרופות חדשות. בשביל זה אנחנו כאן. נמשיך בצעדים קטנים לשינויים גדולים. את כריסטופר פגשנו לפני מספר שנים, באחד הכנסים הראשונים שבהם השתתפנו. כבר ברגע הראשון היה ברור לנו שפגשנו אדם מיוחד, חלק ממשפחת הדושן שהלך בדרך מאוד מיוחדת של הסתכלות על החיים, התמודדות עם אובדן, הודיה, והמשך של עשייה ונתינה לאחרים. למרות הקשיים והמכשולים שעבר, כריסטופר בחר להעצים ולמצוא את המשמעות ברגעים הקטנים של החיים.
Hello, my name's Chris Jones, and I'm the father of a young boy who had Duchenne muscular dystrophy. His name was Mitchell, and I'm excited to spend a few moments with you and share a bit about our story. And the message we want to share today is about how to find gratitude and joy and purpose in the in-between moments, and why those little moments in our life matter more than we think. I'm here today because, like you, I've been touched by a child who had a neuromuscular disease. My son's name was Mitchell. He followed me like a shadow on a sunny day. And if we were the same age, I'm pretty sure he would have been my best friend. Now, at the age of 3, Mitch was diagnosed with Duchenne muscular dystrophy. We spent the next several years worried about his health, yet at the same time trying to learn how to live in the moment and let tomorrow be. At the age of 9, Mitch was diagnosed with cardiomyopathy, premature heart failure. Because the heart is a muscle too, we understood what was happening, but we were surprised to see it happening so quickly. Within about a year of his diagnosis of heart failure, he was sent home on hospice to die. And we had the painful privilege of ushering our son to the other side. I think it's safe to say I've been to hell and back. But I'm back, and I have a story to tell. And that story is that joy and happiness can be found in the in-between. My son and family mean the world to me, and they've grown quite a bit since my little boy has left our family. I've seen our kids graduate from high school. In fact, I married my daughter off just last year and my son just last month. My family continues to grow, and we're learning to take advantage of the moments that are before us. But I think the majority of life can be described in one sentence. It doesn't often go as planned, and that's okay. I'm going to share with you, to illustrate a few points, a few slivers of our life between 2009 and 2013. This was a time where there was great unrest and uncertainty, and at times profound sorrow. We'll probably all relate to this on some level, that during our son's diagnosis, particularly in the latter part of his life, we found that isolation began to grow, that the sicker our son became, the harder it was for people to identify with our situation. Certainty about his future began to decline. Medical complications were on the rise, and stress was at an all-time high. Yet despite all of that, against the backdrop of all this difficulty, we found moments and reasons to be happy. We found little points of light that contributed to our joy in life, despite the circumstances with which we faced. You see, happiness is not a destination. It's a journey with 1,000,001 setbacks. It's a frame of mind. It's an attitude. It's a disposition. We'll talk about that in a moment. So if we were to look at the path of life, there's really a path that leads to happiness and one that leads to despair. And I want to talk about the things that contribute to both. The first, I want to talk about gratitude leading to the path of happiness, whereas comparison leads to despair. It's been said that gratitude turns what we have into enough, while comparison is an illusion that robs us of joy. I remember when Mitchell was home on hospice, I was helping him take a bath, And he pointed to his PICC line in his arm, which was pumping medicine into his heart to help him live just a little longer. He said quietly in a soft voice, "I wish I didn't have to have this." He thought a moment, and then he said, "At least I'm alive." And in that moment, I learned a profound lesson that gratitude, despite our circumstances, contributes to happiness. It's been said that gratitude is not only the greatest of virtues, but the parent of all others. Another great quote, happiness doesn't depend on what we have, but it depends on how we feel toward what we have. We can be happy with little and miserable with much. I remember taking Mitchell into the cardiac intensive care unit when he was first diagnosed with acute heart failure. We were stressed and we were worried about his health and his future, and we didn't know what was happening. And we went down to the basement of the hospital where they have a gift shop, and we got him a little teddy bear to help comfort him during a time of great unrest and uncertainty. And in the store, I noticed a coin that had the word gratitude inscribed on it. And I thought, what a beautiful reminder to practice gratitude in the very moment. I was grateful that I even still had my son with me. And so I learned after that to continue to practice gratitude while the sun is shining, because when the storms of life come, gratitude becomes natural and instinctive on our part. You see, it's not joy that makes us grateful, it's gratitude that makes us joyful. The next step toward happiness is learning to recognize the moments, and particularly the moments in between, as compared to focusing on things and stuff. I remember taking this photo of Mitchell and Natalie in my office, and Mitchell was worried about something, and she just grabbed his sweet little face, kissed his forehead, and gave him a hug. And I took a photo of this with my phone because I didn't want this moment to slip by. And I learned early in my journey with my son to collect moments, not things. You know, if we compare the high points in our life, like the theme park trips and all the adventures and vacations we might ever dream to go on, as compared to the ordinary things in life, we would think that the theme parks and all the great adventures are the things that lead to happiness, but they don't. And I'll make an argument here in just a moment, how the most ordinary things in life are where the most beautiful things exist. We did take our kids to theme parks. We enjoyed them. We had fun at them. No problem there. We enjoyed it very much. But those were just momentary experiences. The moment the theme park was over, the joy was over for the most part. They weren't things that lasted. I'll tell you something that did last. We took our kids to Mexico to go see the ancient ruins of the Incas and the Mayas. And it was beautiful. We were in tropical weather. It was just amazing. And our kids enjoyed the ocean, the warm water. And I remember at one point, very early in our experience at the beach, Mitch flopped to the ground on the shore and said, "I'm so tired. I can't do this anymore." And all the other kids were running around like they could have gone on forever, but Mitch was too tired. So Natalie takes Mitch out into the water and begins to hold him. And they just start having a little conversation. I'm out on the beach with my big lens and taking these pictures with them, completely unaware. And here Natalie is comforting my son and giving him a moment so that he could enjoy the water and feel safe. This was the most beautiful experience of the entire trip. Yet there were many others a lot like them, but this was my favorite. So if you were to compare the two experiences, one, this extravagant vacation, or the ordinary moments, this in between, I'll tell you, that's what I weigh. This has the most weight and value to me. It's the little moments in between. And we had lots of those on the trip that I was grateful to recognize in the moment. Another example of where sometimes the big fun things aren't so much the great things is we had a bounce house that your kids would use in the summer, and we'd fill it up with water, and they'd go sliding down the water slides. And it was fun. Yeah, our kids really enjoyed it. But I discovered something. About an hour and a half later, it got really quiet up in our backyard. And I go out there, and what I see is a little fort created that my kids made. And there in the shadow of this summer fort are my three boys having fun. And they had spent the entire afternoon in this little thing. It was adorable, sort of, because here's the thing. Here's the math, right? The Costco bouncy water thing that we bought for $1,000 US, you know, as compared to a little cheap swimming pool and PVC pipe and towels. My kids had more fun with the $20 thing than they ever did with the expensive thing. And we were glad to have both experiences, no doubt about it. But it's funny that our kids don't need extravagant things. They just need time, attention, and maybe a little imagination. And imagination takes them a lot of places. I remember my boy, for his birthday, got a little Star Wars gift, and he jumped inside the box, and that was his fort for the day. Mitch did the same thing. He would grab old cardboard boxes with tape, and we began to construct forts out of cardboard boxes as a tiny child. Just before he went on to hospice, Mitch asked his mom, hey, can I sleep in that big cardboard box? We just finished our basement. This was the box for our bathtub. And he wanted to sleep in this for the next couple of weeks. So for over a month, my little boy slept in our living room in the front of our house in a big cardboard box, and that was his fort. And every night his mom would tuck him in, and it was really cute. He was able to enjoy life and make moments of his own. We have a philosophy at our family that we're always down to build blanket forts. And what that means, we're just always excited to create forts of any kind, whether they're out of cardboard boxes or blankets or pillows or all of it together. And so we did some Google searching and found some amazing examples of how you can build really extravagant box forts. And so if you're in a place to do this, I recommend it. I wished I would have done stuff this great when I was a kid, when I had younger kids. But what I want to say is pay attention to the in-betweens, because the very best moments in your life exist in the in-between. 95% of life is ordinary. The monotony of life consumes most of it. Yes, we have high points like a fun vacation or a difficult diagnosis is a low point, but the majority of our life is the mundane. And that, my friends, is where the magic happens. That's where we have those tiny conversations with grandparents and begin to notice those between the cracks of things. The dancing in the kitchen spontaneously, the teasing of our siblings, the lessons playing the piano, or reading books at bedtime, the long walks with our kids, or helping a sibling who struggles, the snow adventures, the adventures our grandparents weave, the little experiences we have with our best childhood friends, the bedtime conversations, learning to do new things, watching our grandparents do crazy things. Those conversations, these little moments in between, are what matter the most. I'm so glad I took this photo. I took the time to recognize this little in-between moment with my wife, pregnant with my youngest son, helping Mitchell and Ethan, my two boys at the time, swing on our swing set. And we were so worried because we already knew our son, Mitch, was diagnosed with DMD, but my wife took a moment to just be in the moment with my kids. And to me, that's a beautiful, beautiful memory. I'll never forget these tiny in-between moments, these conversations, these little crazy adventures. But the truth about parenting is that the days are long, but when you think about it, the years are short. I remember taking my kids on a quick walk with my wife. They all jumped on bikes and scooters, and we all went out on a summer evening. And I was writing an essay not too long ago, and I wrote this. I said, I miss everything in between. I miss everything that was ever routine. What I've learned in my own grief journey is that the things I miss the most aren't the fancy vacations, it's the routine things. It's the things that I always took for granted were the things that I want back. And if that's what I want back, then that's the stuff I should be investing in right now for the future. That's what I've learned. Relationships are the path to happiness. Isolation, not so much. Harvard University did a study not too long ago, and they said, what if we could watch people's lives like a movie through time? What would we discover about human happiness? They tracked the lives of over 700 people for over 80 years. Since then, they've added another 2,000 of their children to the study. What they did is they took groups of people from different socioeconomic backgrounds and began to watch, what does circumstance have to do with happiness? And what are the other factors that contribute to happiness? And what they learned was this. When the study began, nobody cared about empathy or attachment. But as it turns out, the key to happiness is relationships, relationships, relationships. The 3 big lessons this Harvard study came out with was social connections are really good for us. The quality of relationships matters. And good relationships are not only good for our bodies, they're good for our brains. Good relationships keep us happier and healthier, period. That's what the data presents. Relationships are the key to happiness. Lastly, let's talk briefly about meaning and purpose as compared to circumstance. When we focus on meaning and purpose, we find happiness. When we focus on our circumstance, that almost inevitably leads to disappointment and frustration, because guess what? Life never goes as planned, right? So there's a happiness paradox. Viktor Frankl said that it's the very pursuit of happiness that thwarts happiness. That, my friends, is a deep, deep truth. If you think back on what made you happy in the last week, what is it? Think back on what made you truly happy. And if you need to think back a month or a year from now, think back on what made you the happiest. You see, here's a diagram I put together that would compare different types of things that lead to happiness. There's things that are pleasurable, that are temporary moments that bring us some measure of happiness. There are things that we're passionate about that evoke strong emotions. And then lastly, there's things of meaning and purpose, things that transcend material things. In matters of happiness, I think sometimes we can confuse short-term pleasures for long-term fulfillment. Let me show you an example. Things that are pleasurable are like vacations, parties, camping, food, video games, reading, and other things. Things that your happiness ends the moment that thing ends. Then there are things that we're passionate about, like photography and running or writing or sports or anything else that you love to do that you're particularly passionate about. And then lastly, there are things that have meaning and purpose, like parenthood or family or service or spiritual discovery or other things that are deeply meaningful to you. These are sources of happiness. And what I've learned is I love photography and camping. I love going out. I've traveled the world and taken lots of photos, and camping's been lots of fun. And I've learned to take my kids with me. I've learned to involve them in the things that I love, and I'm able to make connections. In fact, we did this just a few years ago in 2017. We took a photo of the eclipse in Oregon, and that was where we had totality, and it was amazing. This was our favorite photo. Look at all those sun flares. It was really a beautiful experience with our family. My point here is this, that if all we do is spend time doing the short-term pleasurable things, we'll miss out on the deeper, richer things in life. And so there's a way, if we are thoughtful about it, we can connect the things that bring us pleasure, the things we're passionate about, with things of meaning and purpose. So think of things that your family or you love to do and see if you can involve others in that, and you'll find that it'll even be more rich than you imagine. Seek for meaning and purpose first, and happiness will follow. This, my friends, is the path that leads to happiness in my life experience: to practice gratitude, to recognize the moments in between, to invest my time and heart in relationships, and to seek after meaning and purpose. As compared to comparing my life with others, to looking at what things do I have or not have, to being alone, or to be just focused on my circumstance, tend to lead to despair. So my parting thoughts are practice gratitude, practice gratitude, practice gratitude. That's the key. And when you are grateful, you begin to recognize those moments in the in-between. This image right here isn't just an icon. It's actually a photograph I took of my sons as they were on a walk on a fall day. And it was a beautiful reminder to me of those moments in the in-between. My friends, at the end of the day, I want you to look back on your life a year from now or a lifetime from now and be glad you lived the life you lived. And the best way to find gladness and gratitude for living life is to enjoy the moments in between, because that's where we spend most of our time, are the moments in between. Thank you for your time. I hope some of these ideas are helpful as we all go through our journey in life, trying to find meaning and purpose and joy.
פרופסור אניקה ארצמה-רוס נחשבת למומחית בעלת שם עולמי בתחום הגנטיקה בדושן. יש לה פינה חמה בלב לעמותה שלנו, וזה לא מובן מאליו, וזו הזדמנות מצוינת להודות לה על כך. דושן היא מחלה מורכבת, והיעדר הדיסטרופין משפיע על מעגלים רחבים בגוף. אנחנו יודעים היום שלא תהיה תרופה אחת שתרפא את המחלה, אלא יהיה צורך בשילוב של אסטרטגיות. חלקן יטופלו בגורם הגנטי, חלקן יפחיתו את התהליך הדלקתי, וחלקן יפעלו להאטת תהליכים אחרים המתרחשים בגוף בעקבות המחסור בדיסטרופין. חברות המחקר שמשתתפות במושבים הקרובים בכנס יציגו מחקרים מאסטרטגיות שונות. חלקם מיועדים לחולים שעודם הולכים, וחלקם לחולים שאינם כבר יכולים ללכת. זו התקדמות משמעותית מאוד במחקר שחלה רק בשנים האחרונות. אנמיקה תפרוס בפנינו את אסטרטגיות הטיפול השונות.
Hello, I'm Annika Archmares. I'm from the Leiden University Medical Center, and I'm very sorry that I cannot be with you in person today because, well, if you know me, you know that I love Israeli food. I also love your country. I love the weather that's often much better than the Dutch weather. So I'm very sorry that I cannot be with you in person. But it's great that I at least can contribute in this way to your virtual conference. And of course, congratulations to the 10-year anniversary. And I would like to commend you also for all the nice work that you have achieved in the past 10 years. So what I will do in the next 10-15 minutes is to introduce Duchenne Dystrophin and also the way that different therapeutic approaches can try to improve the pathology of Duchenne. So these are my disclosures. And Duchenne is of course caused by lack of dystrophin. And dystrophin normally stabilizes muscle fibers when you use your muscle. And of course, there's a lot of forces that come on the muscle when you use them. And dystrophin prevents these forces from causing damage. And it does that by connecting the inside of the muscle fibers to the outside of the muscle fibers. You can compare that to this carabiner system. And the way that the carabiner system works is you don't need this to go rock climbing. You need this to avoid damage when you make a mistake. Well, this is the same thing with dystrophin. You don't need dystrophin to use your muscle. You need dystrophin to avoid that things go wrong. And if you use your muscles without dystrophin, things go wrong all the time. And we know this because Duchenne patients don't have dystrophin. And even when they use their muscle in a normal way, they continuously have damage. And that is because this linker function is lost. Now there's also Becker muscular dystrophy, and here patients make a shorter protein. But what you can see is that with dystrophin, the connection to the rope is lost. So if you fall, you crash. In Becker patients, both connecting domains are there, only the elastic bit in the middle is shorter. That means that if you fall, it's uncomfortable, and you may bump against the rocks, but you will not crash. And this is the same in patients. Duchenne patients have a very severe disease. It's progressive, while Becker patients have a less severe disease that is less progressive. And I don't want to trivialize Becker. It's still a severe disease, but compared to Duchenne, it is less severe. So now we can go into what happens when there is no dystrophin. And the first thing I already mentioned, if there is no dystrophin, patients will get muscle damage all the time when they use their muscle. Now, normally, having muscle damage is not a problem. Also, healthy people can sometimes have muscle damage, but that is then repaired. However, this is a process that happens sequentially. So when there's muscle damage, first you have inflammation, which then clears away all the rubbish, and then you have the repair. Just like if you refurbish your kitchen, you take out the old kitchen first, and then you start building your new kitchen. You're not gonna put in new cupboards when the old ones are still there. So this happens in the same way with muscle repair. However, because Duchenne patients have damage all the time, there's always inflammation somewhere in the muscle. And that means that even the areas where there should be repair, there is no repair because the inflammation inhibits the repair. So that's one problem. There's an inhibition of the repair. There's also chronic inflammation because there's damage all the time. And when there's chronic inflammation, this will mean oxidative stress, and this will mean that fibrosis is formed. So rather than having new muscle, you get scar tissue formation. And once there is fibrosis, so once there is this scar tissue, this tissue produces factors that further inhibit repair. So you can already see that things go wrong continuously. Now, another thing, if there is no dystrophin, is that there's less blood flow. And if there's less blood flow, that means more muscle damage, because when you use your muscle, you need the nutrients, you need the oxygen. When this is not there, you get more muscle damage, you get oxidative stress. And again, you get more of this inflammation, fibrosis, and less repair. Now, finally, there's too much calcium ions in the skeletal muscles due to lack of dystrophin. And this means that there's less energy production because the power generators of the cell, the mitochondria, are very sensitive to how much calcium there is in the muscle fibers. Because there's less energy, there's more oxidative stress, there's again more damage. So all these pathways jointly contribute to a continuous loss of muscle tissue and loss of muscle function. And this is what happens in Duchenne. In Becker, this happens as well, but at a much slower pace. So then how can we address the pathology? How can we interfere with this? Now, one thing is, of course, what you can say, let's target inflammation. So we see here inflammation. If we would reduce inflammation, we expect that there's more repair. We expect that the oxidative stress will be less, that there will be less fibrosis. We're not going to counteract everything, but at least we may slow down part of the pathology. Well, suppressing inflammation is, of course, already done. So almost all patients take corticosteroids. Corticosteroids work in multiple ways, but one of the things that they do is that they inhibit inflammation, and that is probably their main reason for slowing down disease progression in Duchenne. Corticosteroids are not approved for Duchenne. They are used off-label. So we know that they inhibit inflammation, and we know that they can slow down disease progression. They're in the standards of care, but they're not specifically registered for treating Duchenne. So they're used off-label, but by almost all patients. The problem with chronic use of corticosteroids is, of course, all the side effects. And that's why a new drug is in development called vamorolone. And the hope is that this will do the beneficial aspects of the corticosteroids, but with much less side effects. And whether that is indeed the case, we don't know. It's in trial so far. Studies are encouraging, but more work is really needed to see whether it can give the same benefit and whether indeed there are less side effects. So this is being studied currently in studies, clinical trials in Europe and the US. Then there's another compound called adasalonexant, and that is very specifically targeting the inflammation. And unfortunately, we heard last week, last week when I taped this, so end of October, we heard that unfortunately the clinical trial did not show benefit. So this development is stopped, and this again shows that the steroids do more than just acting on the inflammation. So unfortunately, this development has stopped. And we'll have to wait and see what the vamorolone brings. Hopefully, it will do the same as the steroids without the severe side effects. Now, of course, there's other areas you can interfere. You can also interfere with the oxidative stress and the fibrosis formation. And oxidative stress, I mean, antioxidants, we all know them. Green tea has antioxidants. The problem is that if you really want to have an impact, you would have to drink liters and liters and liters of green tea. Well, that's not going to work. So there are more potent antioxidants. And these have been tested in Duchenne as well. One of them is called idebenone. And also here, unfortunately, in October, we learned that there was no benefit in a phase 3 clinical trial. So this development, unfortunately, also has stopped because this was not sufficiently efficient to slow down disease progression in Duchenne. There's also another compound in clinical trials that's called pamrevlumab, and this is an anti-CTGF antibody, and so CTGF is a very important player in fibrosis formation. So the hope is that if you inhibit this, you will slow down fibrosis. A phase 2 clinical trials ongoing in the US. We don't know yet whether this will have an effect or not. We will have to wait and see for that. Now, the challenge is that if you target just one part of the pathology, all the other things will continue. So then, of course, it would be better to target different areas of the pathology. Well, there's one compound that aims to do that. That's called givinostat. Here, the repair should be improved, inflammation should be reduced, and fibrosis should also be reduced. Whether that is really the case, we'll have to wait and see again, because this is currently tested in a phase 3 clinical trial. So we'll have to wait and see whether indeed this has effects on repair, inflammation, and fibrosis, and whether also slows down disease progression. So we'll have to wait for that. Now, of course, all these pathways occur because there is no dystrophin. So if we could restore dystrophin, all these downstream pathways would be slowed down. So that is maybe a more effective way. And there's different ways that therapies that are in development now try to address this. There's Axon Skipping, there's stop codon retrieval, and there's gene addition, and all these three are in clinical trials. And there's also genome editing that is in an earlier stage, and that will be covered in another lecture. So I'll now explain Axon Skipping, stop codon retrieval, and gene addition. Starting with Axon Skipping. So we have the blueprint of all the proteins that we make in our genes, including the dystrophin gene. And the genetic code of the protein is dispersed over parts of the gene. And these things are called exons. So the exons contain the genetic code. In between, there's parts of the gene that do not contain the genetic code, don't code for protein. They are called introns. Now, when a protein needs to be made, first a temporary copy of the gene is made, called RNA, and that contains both the protein coding information and the non-coding information. And before this code is translated into protein, these non-coding pieces need to be removed. So all the exons, the coding containing information pieces, are linked together in a process called splicing. You then end up with your messenger RNA containing only the code, which is then translated in the cell into protein. And this is currently happening for all our proteins in all our cells. Now, what you could see is that these exons are like puzzle pieces and they all fit together. What happens in Duchenne patients is that most often one or more exons are missing, and that then the puzzle pieces don't fit. So you see here, exon 48, 49, and 50 are missing, and 47 and 51 don't fit. And then we say the reading frame is disrupted. The code becomes unreadable because the cell can translate until 47, and here the code becomes unreadable, so translation stops. And this is why the dystrophin is not functional, because only the beginning part is made, and this link, the important last domain that needs to link, is not there. So these are the Duchenne mutations. Now, in Becker patients, what happens also often, one or more exons are missing. You see here 48, 49, 50, and 51 are missing, but now 47 fits to 52. So the code is readable, the frame is not disrupted. So that means that the translation can continue. There is a little bit missing here in the middle because some of the coding pieces are missing. However, the crucial part of the dystrophin at the beginning and the end, these are there. And this is why Becker-like dystrophins are partially functional. Now, the idea of exon skipping is to allow Duchenne patients to make a Becker-like dystrophin. And again, here we have this temporary copy with the exons and the introns, so the coding parts and the non-coding parts. And what we see is 51 doesn't fit to 47. We've seen that already. And what we also know is 47 would fit to 52. So what we want the cell to ignore this one, exon 51. And we can do that with antisense oligonucleotides, or AONs, short pieces of modified RNA that bind specifically, in this case, to exon 51, hide it. Now 47 is joined to 52, the code is restored, and the cells can produce a Becker-like dystrophin. So where are we at with exon skipping at the moment? Well, first of all, this is a mutation-specific approach. So depending on your mutation, the size, the location, you need to skip specific exons. There's currently 3 AONs approved, 1 for 51 and 2 for 53, and they are approved in the US and Japan only. This approval is based on dystrophin restoration and very low amounts of dystrophin. So less than 1% to about 5%. And I always say some dystrophin is better than none, but with these low levels, the question really is this enough to slow down disease progression? And that is something that the companies that develop these AONs, they still need to show. They need to show the regulatory agency that indeed this has functional consequences, and the clinical trials to do this are currently ongoing. Now, what is clear is that we need to improve these AONs. There's room for improvement. So there's also work ongoing on trying to make better chemistry, to improve the delivery to skeletal muscles. And this work is ongoing, but the problem here is that these are using new chemistries, and then of course we don't know whether these will be safe in humans. So that is then the thing that will still need to be discovered. Now, the second thing I will talk about is stop codon read-through, and this applies to very specific mutations. So again, here we have this messenger RNA, the code for the protein. It has a start signal, so the translation machinery knows I need to start translating here, and at the end it has a stop, so it knows protein is complete. Now, what is possible is that the code has a very minor change, whereby the code for a protein subunit is now converted into the code for a stop. And this will be used, and then of course only the beginning of the protein is made. However, the rest of the code is still intact. Now, there are compounds that can suppress the use of these fake stops that shouldn't be there, but due to a small mutation are there. And one of these compounds is called ataluren. And again, I want to show here that this only works for these nonsense mutations. So here again, we have the start, we have the stop, and now here, the only change that happens is that suddenly here we have an extra stop. If we have these mutations that disrupt the code, so the frame shift that make the code unreadable, this will not work because the code will still be unreadable. The ataluren doesn't change that, it only suppresses these type of mutations. Now, what is the current state of the art of this ataluren, this stop codon read-through compound? It is conditionally approved in Europe for ambulant patients of 2 years and older. Conditional approval means that every year it's reevaluated. Is it safe and is it still effective until the company acquires enough information to get full approval. It's marketed in several European countries. I know it's marketed also in Israel, but it's not available everywhere. So for instance, it's not yet available in the Netherlands. There's trials ongoing because it's conditionally approved. The company needs to collect additional information to show that this indeed has also functional effects. So there's several trials ongoing. And in addition, the patients that are now treated commercially with this drug are also being followed and data is collected from them. And so far, the results look encouraging. So again, we'll have to wait and see. But for now, at least this is available. Then finally, gene addition. The dystrophin gene is not functional in Duchenne patients. Can we give them a copy of the dystrophin gene that is functional? And yes, we can do that, but the problem is, how do we deliver that gene? Well, for this, we need to use viral vectors because viruses do nothing more than delivering their genetic information to human cells. Now, the problem with viral vectors is that most often they do not infect skeletal muscle. There is one exception, and that is AAV, adeno-associated virus. AAV does not cause disease, so that is a good thing, but it's a very small virus. So it has a limited capacity for how much DNA you can put in. Unfortunately, the dystrophin gene is the largest gene that we know. So what we need to do, we need to make a very short dystrophin. So what several researchers have done, they've generated a micro-dystrophin. So in a minimalistic approach, they have made a dystrophin that contains only the crucial domains. And again, if we have our carabiner, so we know this is important and this is important. So this bit here corresponds to this, and this bit here corresponds to that. And you can see here these 3 microdystrophins. You can see that they have these important domains, and then in the middle, the elastic part, that varies. So there's currently 3 companies, Solid, Pfizer, and Sarepta, and each of them has a slightly different microdystrophin that of course all has these crucial domains. And these are currently tested in clinical trials, and you will hear more about that in this session. So in summary, Duchenne and Becker are both caused by dystrophin mutations. The therapeutic approaches that are used or are in the clinic now for Duchenne are corticosteroids used off-label, etolurin in Europe only for nonsense mutation, and Axon Skipping only in the US and Japan for other mutations. And there's also a lot of therapeutic approaches in development, some that try to address the pathology, and these you can use for both Duchenne and Becker, and some that try to restore the dystrophin, and then it's always a partially functional dystrophin like Becker, so this would only apply for Duchenne. So thank you very much, and if you have any questions, then of course I'm happy to answer them later in the Q&A session.
החלק הבא של ההרצאות יתמקד במחקרים בתחום הטיפול הגנטי. כדי להציג את המחקרים ולהסביר באופן רחב יותר על תחום המחקר שנחשב היום כפורץ דרך, אני שמחה לצרף אלינו את דוקטור טליה דור, מנהלת המרפאה הנוירומוסקולרי במרכז הרפואי הדסה. ערב טוב.
היי טלי, תודה רבה על ההזמנה, תודה להיות כאן, מאוד מרגש להיות חלק מהקהילה הזאת וכבוד גדול בשבילי. אני אתחיל לדבר על התרפיה הגנטית, מה העקרונות של הטיפול הזה, פרופסור אניקה קצת הזכירה, מה הסיכויים שנותן לנו הטיפול הזה, ומה הסיכון שיכול להיות בטיפול הזה. אז כמו שאנחנו יודעים, המחלה קורית בגלל מוטציה ב-DNA, המוטציה ב-DNA עוברת למולקולת ה-RNA, בגלל המוטציה ב-RNA, החלבון התקין לא יכול להיות מסונתז, וכאן אתם רואים מימין את הדיסטרופין התקין, שכמו שאניקה הזכירה, פרופסור אניקה הזכירה, הדיסטרופין הוא מאוד אלסטי, הוא כמו קפיץ, הוא בנוי מהרבה מאוד ספקטרין ריפיץ כאלה שגורמים לו להיות מאוד אלסטי, וזה הדיסטרופין שבחולי דושן לא קיים, ובחולי בקר קיים באופן פחות תקין, כאשר האסטרטגיות טיפוליות במחלה הזאת היא באמת או לנסות לתקן ברמת ה-RNA את התרופות שאנחנו מכירים בארץ, הטרנסלרנה והאקסונדיז, או לנסות לשנות משהו בתהליך של החלבון, והטיפול הדפיניטיבי שכרגע אנחנו נדבר עליו בפגישה שלנו הערב, זה הטיפול בתרפיה גנטית. אז מה בעצם הטיפול בתרפיה גנטית אומר? שלצד הדיסטרופין הלא תקין, אנחנו מחדירים עוד גן נוסף שיהיה בסמוך ולצד הכרומוזומים שיש לנו ממילא באופן טבעי בגוף, גן נוסף תקין, שהוא יחיה לצד הגן הלא תקין שנמצא בגוף שלנו. עכשיו, הכיוונים העתידיים בנושא הקריספרים, שמעתם, אולי ינסו בהמשך לתקן את הרצף של הגן, אבל כרגע אנחנו מדברים על טיפול שייכנס ויהיה לצד ה-DNA. הבעיה העיקרית זה איך מחדירים DNA לתוך התא, והבעיה העיקרית זה החדרת הטיפול. כאשר פה נעזרים בטבע, לוקחים את הווירוס, שהווירוס יודע לחדור לתוך התא, משתמשים בו, מוציאים ממנו את ה-DNA שלו, של הווירוס, ובמקום ה-DNA של הווירוס, שמים DNA של מקודד בסופו של דבר לחלבון הדיסטרופין. וכמו שאתם רואים פה במצגת, הדיסטרופין יהיה לצד ה-DNA הלא תקין בצורה מעגלית, ויתחיל לסנטז את הדיסטרופין שאותו אנחנו רוצים להביא לרקמה. וכאן אנחנו משתמשים בווירוס, וירוס שיודע לזהות את התא שריר, לחדור אליו בעזרת הקפסית. אתם רואים כאן את התמונה הזאת, הכחול זה הווירוס חודר לתוך התא, ויש לנו את המעטפת החלבונית. המעטפת החלבונית לא עוטפת DNA ויראלי. את ה-DNA של הווירוס העפנו החוצה, לא אנחנו, החברות, ובמקום ה-DNA של הווירוס יש לנו DNA שמקודד לדיסטרופין. יש לנו פה וירוס מהודס. הגנים הוויראליים הוחלפו החוצה, ובמקומם שמנו את הגן הרצוי, שבמקרה שלנו זה הדיסטרופין, ולא הדיסטרופין, אלא המיקרו-דיסטרופין, שתכף נדבר על זה. אני רק רוצה להזכיר שהטכניקה הזאת כבר קיימת במחלות וכבר אושרה לטיפול, גם במחלות ניוון של הרשתית, של העין, וזה פיתוח שגם לקחו בו חלק חוקרים מהדסה, וכולכם שמעתם כמובן על התרפיה הגנטית ב-SMA, שיש לנו כאן את הגן של ה-SMN, שלא קיים תקין אצל החולים, הוא עטוף בווירוס, והווירוס הזה, יחד עם הגן התקין, מוחדר לחולים עם תוצאות מאוד טובות. אנחנו היום תכף נשמע 4 מרצים שונים מ4 חברות שונות, שכל אחת תציג את הפיתוח שלה, כאשר השוני בין החברות השונות הוא למעשה הבחירה השונה של המעטפת החלבונית, והתכנון של הדיסטרופין. כי כמו שאתם רואים, הגן של ה-SMN הוא קטן, הוא רק 8 אקסונים, והדיסטרופין הוא גן ענק, הוא 79 אקסונים, הם לא יכולים להיכנס לתוך הקפסיד החלבוני של הווירוס, ולכן צריך לקצר אותם. כל חברה תכננה איזשהו רצף דיסטרופין אחר, ובחרה וירוס אחר שבעזרתו היא תעטוף את המיקרו-דיסטרופין, וככה תצליח, ככה בתקווה זה ייכנס לתוך התא השריר ויתחיל לבטא את הדיסטרופין הקטן. ככה שבסופו של דבר אנחנו לא מקבלים את כל החלבון האלסטי, התקין, כמו שאניקה הסבירה קודם, כאילו שמחזיק לנו טוב בצורה אלסטית את שתי הקצוות, אלא חלבון שהוא יותר קצר ויותר קטן. אני רוצה לסכם ולהגיד שבאמת התרפיה הגנטית נותנת לנו כאן אופק חדש של באמת סיכוי לזה שנצליח לקבל חלבון ברקמה של השריר אצל ילדים שכרגע אין להם את הדיסטרופין בגלל המוטציה, ובאמת זה טיפול שאם הוא יעבוד הוא טיפול חד פעמי, הזרקה בודדת ונותן פתרון בתקווה לאורך זמן. כמובן שיש גם סיכונים, החדרה של גן חדש לתוך התא, אנחנו לא בוודאות בטוחים מה יכול לקרות, אבל שימוש דווקא ב-AAV שהוא קטן, והוא וירוס שלא נכנס לתוך ה-DNA של התא. הוא לא עובר אינטגרציה בתוך הגנום, הוא חי מחוץ לגנום, ולכן הוא לא אמור להיכנס לתוך המטען הגנטי שלנו, ואנחנו, בעיה נוספת שיכולה להיות, זה באמת התגובה החיסונית הסוערת, שיכולה להיות לווירוס, כמו שאנחנו רואים, מה קורה כשגוף מגיב לווירוס, וגם פה ה-AAV, הווירוס שנבחר, הוא וירוס שבאמת מעורר הרבה פחות תגובה חיסונית, ונותן לנו סיכוי באמת לטיפול טוב ותקין, וכולנו תקווה שבאמת עם הכניסה של הפאזה השלישית למחקרים הקליניים נראה תוצאות טובות.
תודה רבה, דוקטור דור.
סיימתי את החלק שלי.
תודה רבה. אנחנו נעבור עתה להרצאות של חברות המחקר, סרפטה בשיתוף ראש, סוליד-ביו, ג'נטון ופייזר.
Hello, I'm Dr. Alex Murphy, and I work for Roche Pharmaceuticals as a medical director. And I'm here today to talk to you about microdystrophin and gene therapy. This second slide is just our disclaimer, and it's saying that we developed this presentation jointly between Roche and Sarepta pharmaceutical companies, and it was finally approved by Roche. This is all part of our partnership between Roche and Sarepta. So, neuroscience is a major part of research and development at Roche. Our goal is to pursue groundbreaking science to develop new therapies to improve the lives of people living with chronic neurological diseases. Roche's aspiration in neuroscience and rare disease is to preserve the innate brilliance inside us all, what makes us unique as individuals, beyond any disease or condition. It is this aspiration and commitment to develop transformational therapies that has led to Roche investing in gene therapy for rare disease. In December of 2019, Roche partnered with Sarepta Therapeutics to collaborate to improve the lives of people living with Duchenne. Together, we will reach more patients with Duchenne faster than we could on our own. In order to talk about our trials, it's important to first understand what gene therapy is. Gene therapy focuses on the transfer of genetic material to a person to treat a genetic disorder. The aim of transferring genetic material is to compensate for genes that are not working correctly, or that could work better, or to make a beneficial protein. Gene therapy aims to promote sustained expression of the therapeutic gene, or as we call it, the transgene, at a level high enough to be considered therapeutic and make a difference to the patients. Clinical investigation using gene therapy first started way back in 1990, and now more than 2,500 clinical studies have been initiated for a broad range of rare and neurological disorders. In order for the potential of gene therapy to be realized in any therapeutic intervention, three essential components are necessary: the vector, the promoter, and the transgene. The vector refers to the adeno-associated virus, or AAV, that's used to carry and deliver the transgene to relevant tissues. The vector is also the main component that interacts with the host immune system, and thus it is believed to be the major driver of the safety profile for the therapy. The promoter controls which cells express the transgene delivered by the vector. The promoter also controls how much transgene the target cells will produce. In the case of Duchenne, the aim is to get them into muscle cells. The transgene is, of course, a critical component of the whole thing. It intends to produce a functioning version of the protein of interest. For Duchenne, that would be dystrophin. Dystrophin itself is one of the largest genes known to man, and it doesn't fit within a viral vector. Therefore, microdystrophin has been developed and engineered so that it includes the most critical parts of the dystrophin while still fitting within the vector. When evaluating any gene therapy as a potential treatment for a genetic disorder, we must ask ourselves several questions, and these are evaluated in clinical trials. Firstly, what is the safety and tolerability of the treatment? And that's the same for any medicine. It's important to establish that first. Secondly, is the transgene in the target cells? Is the desired protein being made? And is it in the right place? These are all assessed through clinical trials using Western blot and laboratory techniques such as that. Finally, do patients show an improvement in function? And this is measured by various assessments, including physical examinations, usually led by a physiotherapist. And these include assessments such as the North Star assessment. So this brings us on to the SRP-9001, which is the medicine in question. The first trial that was looking at this study drug was called Study 101. It's an open-label, single-dose trial. This means all participants received the SRP-9001. This was a phase 1 or 2A trial, meaning it was an early phase trial, that enrolled 4 subjects with Duchenne at the age of 4 to 7 years. The primary objective of the study was to evaluate safety and tolerability of SRP-9001. All patients were negative of the antibodies to the viral vector. All patients were on a stable dose of corticosteroids for at least 3 months prior to gene transfer. The second trial seen here is Study 102. It's a double-blind, randomized, placebo-controlled phase 2 study. This means that neither the patient nor the investigator knew whether they were actually receiving the SRP-9001 or a placebo, which is a medicine not expected to have any effect, such as saline. 41 patients were enrolled. All are dosed and are in the follow-up period. Crossover has now started while still in the double-blind phase, meaning patients initially in the placebo arm are now being dosed with SRP-9001. Future trials support our goal to expand our eligibility criteria across different studies, with the first slated to begin soon. Our next studies include trials 103, which is a phase 1 trial, and a phase 3 trial, with age criteria similar to our studies of 101 and 102. In addition, we're currently planning a trial which will involve individuals who are non-ambulatory. It's important to note that with some medicines, as we develop them, small amounts are at first made using a particular method. When you want to make larger amounts of the medicine and scale up, it's important to show that the new way of making the medicine, called the commercial material, results in the same effect, and that the scaling up process hasn't meant it's less safe or effective. Study 103, which is now called Endeavour, is an open-label systemic gene delivery study evaluating the safety of and expression of SRP-9001 in 10 subjects with Duchenne muscular dystrophy at 12 weeks post-infusion, using this commercial process material. It's considered a phase 1 study and is thought of as a bridging study to assess if we get the same outcome with the commercial supply as we did with the clinical supply, the smaller volume, in our previous SRP-9001 studies, which is 101 and 102 I've just spoken about. This is separate from our global placebo-controlled phase 3 trial. Finally, we would like to thank the patients and their families who take part in clinical trials, as well as patient advocacy groups and caregivers who provide valuable input into the design of clinical trials and ongoing research. Thank you very much for inviting me to speak today, and please enjoy the rest of the conference.
ערב טוב, אני מציג הערב את תוכנית המחקר של הטיפול הגנטי של חברת פייזר. זה מחקר ששלב 3 שלו התבצע בארץ, בשניידר וגם בבית חולים הדסה. פייזר היא חברה תרופות ותיקה ומוכרת, היא פרסמה גם לאחרונה בעקבות החיסון לקורונה, והיא מתגאה בכך שיש לה תוכנית שלמה לטיפול גנטי, שכוללת החל ממחקר בסיסי למחקר קליני, וכלה במספר בתי חרושת לייצור הוקטורים והטיפול כאשר הטיפול יוכח כיעיל. החברות השונות שונות במספר פרמטרים, אחד בנגיף, הווירוס עצמו שמכיל את הגן שבו מטפלים, ובחברת פייזר זה AAV9, הוא מוכר, הוא כבר בשימוש כתרופה, למשל בזולגנזמה, שזה טיפול ב-SMA, שמאושר גם על ידי משרד הבריאות בארץ. יש פרומוטור, משהו שמוביל את הווירוס עם הגן ספציפית לתוך תאי השריר, ובתוך הווירוס הזה, כפי שכבר שמעתם בהרצאות הקודמות, לא יכול להיכנס הדיסטרופין שלם, ויש מיני דיסטרופין ספציפי לכל חברה, ובשיקופית שלפניכם יש את המיני דיסטרופין הספציפי של מחקר זה. אני רוצה תחילה להציג את התוצאות של הפאזה הראשונה, במחקר שנעשה בבני אדם, לטיפול באותו טיפול גנטי של חברת פייזר, ובמחקר הזה שהשתתפו בו 9 ילדים, היו 2 מינונים, מינון נמוך, מינון גבוה. למעשה מטרת פאזה אחת היא למעשה בטיחות, אנחנו נשתתף בהמשך במשהו שיוכיח יעילות, אבל אנחנו נראה שגם במחקר הזה, שהוא רק על 9 ילדים, אפשר גם לראות מדדים מסוימים של יעילות כבר של הטיפול. החולים שהשתתפו במחקר הזה, נלקחו להם ביופסיות לפני התחלת הטיפול, אחרי חודשיים ואחרי שנה. דרך אגב, במחקר שיערך בארץ לא ילקחו ביופסיות שריר, ואני אראה לכם תכף את התוצאות ואת הקשיים שהיו במחקר. אז כאמור, המחקר היה ב3 מרכזים בארצות הברית, 9 משתתפים, גילאים 6 עד 12 וחצי, קצת יותר מהגילאים של המחקר שלנו, ילדים עם דושן, שלא היה להם נוגדנים כנגד ה-AAV, כנגד אותו וירוס נשא. אפשר לראות, וזה אולי משהו שבהחלט מעניין, שרק לכ10% מהילדים היו באופן טבעי נוגדנים כנגד הווירוס, מה שאומר שלרוב הילדים באופן טבעי אין, כך שהסיכוי להיכלל במחקר הוא טוב, או לא להיפסל מסיבה זו. התוצאות של המחקר, ראשית נדבר על נושאי הבטיחות, ומבחינת הבטיחות אפשר לראות שהטיפול IV ניתן בהצלחה, לא הייתה פגיעה כבדית, אבל היו מעל ל-40% תופעות לוואי, בעיקר תופעות כמו בחילות, הקאות, חוסר תיאבון, חום. ב3 מתוך הילדים היו להם תופעות שנחשבו כרציניות, serious adverse events. אחד מהילדים היה לו הקאות חוזרות עד כדי התייבשות שהצריך טיפול בנוזלים ובטיפולים כנגד בחילה. ילד אחד היה לו פגיעה כלייתית עם מה שנקרא המוליטיק אורמיק סינדרום, שהצריך אפילו לתקופה מסוימת המודיאליזה וטיפול בתרופה שנקראת אקוליזמאב, וילד שלישי היה לו טרומבוציטופניה, ירידה בתסיות הדם, שהצריך מתן תסיות וגם כן טיפול באותה תרופה שנקראת אקוליזמאב, ואנחנו נדבר על התרופה הזאת טיפה בהמשך. מבחינת התוצאות, נבדקה, בגלל שעשו הרבה ביופסיות, נבדקה רמת הדיסטרופין, ואנחנו יכולים לראות שבילדים רגילים שאינם חולים יש להם דיסטרופין גבוה יותר, והממוצע שלהם הוכרז כ100%. בקר, הממוצע שלהם היה משהו בסביבות נגיד 30%, והממוצע של ילדים עם דושן היה בסביבות 2%. אנחנו יכולים לראות שבמינון הגבוה, אחרי כשנה, אנחנו מגיעים לריכוז של דיסטרופין, ביטוי של דיסטרופין, שהוא בערך מעל 50%. צורה שנייה להסתכל על התוצאות, אנחנו בודקים איזה אחוז או כמה אחוז מהסיבים נצבעו כאשר אנחנו מפעילים נוגדנים עם צבע שמראים שיש דיסטרופין בממברנת התא, ושוב אנחנו רואים שאחרי שנה במינון הגבוה, אנחנו רואים משהו בסביבות ה-50%. אם אנחנו נסתכל על התפקוד, נורסטאר, משהו שאתם מכירים מהמרפאות הרב-צוותיות שלנו, אם זו העקומה הממוצעת, העקומה הגאוסית של תפקוד הנורסטאר של ילדים, מה קורה במהלך שנה, אז הממוצע הוא ירידה מסוימת בנורסטאר, אנחנו רואים שרוב הילדים הם בכיוון הזה שיש להם או אותו דבר או שיפור, ולמעשה יש ממש, אפילו במספר הקטן הזה, סטטיסטית שיפור בנורסטאר משמעותי מאוד אל מול הקבוצה של הפלצבו. גם מימד נוסף, שבו אנחנו בודקים את השומן ב-MRI של השריר, יש, זה נקרא דיקסון, שאנחנו יכולים להדגים את השומן בשריר, ואנחנו רואים שבמינון הגבוה, בניגוד למה שאנחנו מצפים שתהיה החמרה או עלייה ברמת השומן, הייתה אפילו ירידה ברמת השומן כחלק מהטיפול. כאמור, כל התוצאות האלה הן תוצאות ראשוניות על מעט ילדים, אבל בהחלט מראות מגמה מאוד מאוד חיובית. אנחנו הולכים לעשות ניסוי גם בארץ של פאזה 3, יהיו 99 ילדים ברחבי העולם, 2/3 מהם יקבלו את הטיפול בהתחלה, ושליש יקבלו פלצבו, ואחרי שנה זה יתחלף, אלה שקיבלו פלצבו יקבלו את התרופה, ואלה שקיבלו תרופה יקבלו פלצבו, זה כמובן מה שאנחנו קוראים דאבל בליינד, לא אנחנו ולא החולים נדע מי קיבל את הפלצבו ומי קיבל את התרופה, והמעקב יהיה למשך שנה לראות תוצאות, ובמשך לזה 5 שנים לראות תופעות לוואי של הטיפול עצמו. קריטריונים להכללה במחקר, ילדים עם דושן, גיל 4-8, שימוש במינון קבוע של סטרואידים לפחות 3 חודשים, יכולים ללכת 10 מטר, נורסטאר בביקורת הראשונית בין 16 ל-30, ההורים מבינים ויכולים לחתום על המחקר, ושימו לב, צריך להיות חיסונים מלאים, לפחות 3 חיסונים נגד זיהומים פנאוקוקליים, פרוונר, ו4 חיסונים נגד המופילוס אינפלואנזה, כלומר חיסונים מלאים לפי פנקס החיסונים. מי לא יוכלו להיכלל? מי שקיבל טיפול גנטי קודם, אני חושב שאין לנו כאלה בארץ, ילדים שבחצי שנה האחרונה קיבלו תרופות שמשנות את חלבון הדיסטרופין, כמו אקסון דיס או טרנסלרנה, תרופות מחקר אחרות חודש לפני הביקור, ילדים שלא מסוגלים טוב לשתף פעולה, כך שלא נוכל לקבל מהם תוצאות ברורות בחלק התפקודי, ילד שנפצע ולא יכול להשתתף, חיסון חי מוחלש 3 חודשים, חיסון אחר חודש, כל תרופות שמדכאות את מערכת החיסון מלבד סטרואידים הרגילים שאנחנו מכירים, ומורולון לא יכול להיכלל למחקר, זה מדובר על סטרואידים המוכרים ושיש להם אישור כתרופה, ילדים שיש להם מצב רפואי או נפשי כזה שמקשה על המתן. המחקר הוא מאוד אינטנסיבי, הוא כולל הרבה בדיקות, כאשר למעשה לא יהיה ביופסיות שריר, אבל יהיו הרבה בדיקות דם ובדיקות של מעקב רפואי, במיוחד בהתחלה. לפני קבלת התרופה, בגלל שיש אפשרות שיהיו סיבוכים שצריכו טיפול באקוליזמאב, יינתנו עוד 2 חיסונים כנגד מנינגוקוק, שזה יינתן על ידי החברה או על ידינו לילדים שמתחילים את הטיפול. חלק מהילדים, או חלק מהביקורים, הילדים יצטרכו לענוד מכשיר שבודק צעדים, נקרא אקטיגרף. מתן הטיפול עצמו יהיה באשפוז של 24 שעות בבית החולים, והשבועיים אחר כך יהיו בהחלט מאוד אינטנסיביים, כמעט כל יום או יומיים בדיקות רופא דם ושתן, והילדים יצטרכו להיות, כולל לינה, במרחק של שעה נסיעה מבית החולים. אני מסיים בהודעה משמחת ממש של השבוע האחרון, שהטיפול הגנטי של דושן קיבל על ידי FDA אישור של מה שנקרא fast track, מה שאומר שאם המחקר יצליח, ההבאה לכלל טיפול ואישור יהיה מהיר. אני רוצה לסיים בתודה לחולים, למשפחות, לעמותת צעדים קטנים, על הכנס היפה שאורגן, ועל האפשרות לתת את ההרצאה החשובה הזאת על הטיפול הגנטי שעומד להתחיל בארץ. לצוות שלנו שעומל במשך כל השנה הן על מחקר והן על הטיפול בילדים עם דושן. תודה רבה.
For those of you I have not had the pleasure to meet and may not be familiar with Solid-Bio Sciences, I would love to take a few minutes to share our story and ultimately the reason for Solid's existence. I will then hand it over to Joel Schneider, our Chief Technology Officer, and Catherine Clary, our Chief Medical Officer. My son Etani was diagnosed in October 2012. From the moment we received this news, our lives were forever changed. We left our careers and we moved from London to Boston to start a company to not only save our son's life, but all those living with Duchenne. My husband, Dinan, and I, together with some extraordinary individuals, launched Solid in 2014. We are a biotech company focused solely on developing treatments for Duchenne muscular dystrophy. We have employed and partnered with the best and brightest experts in Duchenne and gene therapy. We set lofty goals from the beginning to develop transformative therapies that will improve the lives of all patients living with Duchenne, regardless of age, mutation, or disease progression. And to tackle our goals, we live by 3 core principles. First, we are patient-led. The patient voice is woven into the fabric of everything that we do. We lead with purpose and integrity through listening, learning, and supporting the patient community. We go into every single day with one goal: to improve patients' lives. Second, honesty and transparency. The patient community is a vital stakeholder. Through everything we do as a company, we aim to do as authentically and openly as possible. We believe success is tied directly to the understanding and support of the community behind us. Third, scientific rigor. We're confident that we have the right team in place to bring meaningful therapies to the Duchenne community. We will not stop until we've achieved our goal to advance transformative treatments for those we love and serve. With that, I would like to turn it over to Joel Schneider to provide more information on our lead gene therapy candidate, SGT-001.
Thanks, Annie. As Annie mentioned earlier, my name is Joel Schneider, and I'm the Chief Technology Officer at Solid. First, I'd like to start with a bit of background on our gene therapy candidate called SGT-001. SGT-001 is an investigational gene transfer, a type of gene therapy, of a smaller but functional version of the dystrophin gene called micro-dystrophin. SGT-001 is delivered systemically, enabling it to enter cardiac and skeletal muscles. The SGT-001 microdystrophin is a differentiated construct based on more than 30 years of research conducted by our advisors, Jeff Chamberlain and Dongsheng Duan. It is the only microdystrophin currently in clinical development that's capable of restoring production of nitric oxide, or nNOS, in the muscle, which is important for proper muscle oxygenation and muscle strength. As many of you know, our clinical trial, called IGNITE-DMD, was recently lifted from clinical hold. As part of our commitment to continuously improving our manufacturing processes, we have implemented and shared with the FDA manufacturing changes that remove the majority of empty viral capsids, allowing us to achieve our therapeutic dose target with fewer viral particles. The reduction in the total number of viral particles delivered to each patient is intended to support safe dosing of SGT-001 for the duration of the IGNITE-DMD trial. I would now like to pass it over to Catelyn, who will provide an update on the clinical protocol changes in the IGNITE-DMD study.
Thank you, Joel. As mentioned, my name is Catherine Clary, and I'm the Acting Chief Medical Officer at Solid. For those of you who are not familiar with IGNITE-DMD, I'd like to provide a little background on it. IGNITE-DMD is our phase 1/2 ascending dose clinical trial, and it's designed to assess the safety and tolerability, as well as the efficacy of intravenous SGT-001 in boys with Duchenne. To date, we've dosed 6 patients at 2 dose levels. 3 patients were dosed at 5E13 vector genomes per kilogram, and then 3 patients were dosed at 2E14 vector genomes per kilogram, which is 4 times higher than the lower dose. We have 2 primary endpoints in the study: safety and microdystrophin expression at 12 months, as well as a number of secondary endpoints. Analyzing our muscle biopsies that were taken 3 months after administration from the 2E14 VIGs per kg dose cohort, remember that's the higher dose cohort, we've demonstrated widespread expression of our microdystrophin throughout the muscle in all 3 patients. We also see, very importantly, the restoration of critical dystrophin-associated proteins. Now I'd like to take a moment to address a question I'm sure you have, which is the recent clinical hold. As you may know, a boy who was dosed in the 2E14 VIGs per kg dose cohort last October had a serious adverse reaction, which led to the clinical hold being placed on Ignite DMD. In response to the hold, since safety is paramount to our team at Solid, we did a deep dive into all the data collected to date on all 6 patients to understand what happened. We worked with a number of knowledgeable external experts and did develop a deeper understanding of what transpired in this most recent patient. And ultimately, we developed a clinical risk mitigation strategy, which was designed to improve safety for future dosing. As part of sending this to the FDA, and in order to help decrease the risk of serious drug-related adverse events, we amended the protocol of the Ignite DMD to include the use preventively of 2 anti-complement inhibitors, eculizumab and C1 esterase inhibitor. And we also amended the protocol to include an increase in the prednisone dose in the first month post-dosing. In addition, we're reducing the maximum weight of the next 2 patients dosed to 18 kilograms. This is close to 40 pounds. This reduction, in conjunction with the delivery of fewer viral particles as a result of the manufacturing process improvements, will reduce the total viral load being administered to patients, and we will be able to continue dosing at 2E14 vector genomes per kilogram. It's important to note, and this is very important to us at Solid, that all 6 patients dosed are doing well. All of the SAEs that have been experienced have recovered, and there have been no additional drug-related adverse events up to 30 months post-dosing. Additionally, when we submitted our package to the FDA, we responded to their request for updated safety and functional efficacy data, including the 6-minute walk test and North Star ambulatory assessment for all patients dosed in Ignite DMD. We look forward to sharing these data at a future medical meeting when appropriate. We are working diligently now to complete all the activities necessary to resume dosing, and we expect the next patient will be dosed in the Ignite DMD trial in the first quarter of 2021. For any future updates, please visit clinicaltrials.gov, which is updated, and Ignite DMD clinical trial webpage. Thank you.
Together, we're solid.
Together, we are solid.
We are solid. Together, we are solid.
Good evening. My name is Francesco Montoni, and on behalf of the Geneton, I'm going to present the program related to the AAV gene therapy program for Duchenne muscular dystrophy, and I'm pleased to make this presentation at this Little Step conference on Dושן-בקר muscular dystrophies. Again, my name is Montoni. I work as a principal investigator and consultant in London. I work at the Dubovitz Neuromuscular Center at the UCL Great Ormond Street Institute of Child Health and Great Ormond Street Hospital in London. I have a involvement in a number of gene therapy programs, but also an antisense oligonucleotide program, and my disclosure are indicated in this slide. Regarding the Geneton, this was created by AFM, the French Muscular Dystrophy Association, in 1958 by the mother of 4 boys with Duchenne muscular dystrophy, who felt that not much was available to her at that time. In 1981, the AFM created its first scientific advisory board, and in 1987, the first telethon, a fundraising event, started. This has really been revolutionary because allowed to raise up to 100 million euro each year. And over the years, AFM has invested more than 1 billion euros in innovation since 1987. Therefore, Telethon and AFM are major players in the current biomedical research for rare diseases. In 1990, Geneton was created as a non-profit research and development center dedicated to the treatment of rare genetic diseases with a particular focus on eye, blood, brain, immune system and muscle disorders. In the laboratory at Geneton, there is the possibility to design, produce and test novel medicinal products. Since its inception, more than 30 years ago, the goals of Geneton have changed. In the early 90s, the main focus was that of the identification of genes responsible for neuromuscular disorders. In the late 90s, there was already a focus on gene therapy studies for neuromuscular disorder, and a decade after, the biomanufacturing started, and this in turn led to the identification and the commercialization 30 years after the beginning of the first drug that is now commercially available in the States, in Europe, and in Japan for spinal muscular atrophy, a drug that we know very well. There are, in addition, 7 products at the moment resulted from Geneton research in clinical trials, and another 7 products will be entering in clinical trial in the coming years. In 2014, regarding the specific program for Duchenne muscular dystrophy, Geneton obtained orphan drug designation. In 2020, that is this year, the baseline natural history study started, and I will describe this a little later. And the gene therapy trial protocol has been submitted now to the French authorities. Other important aspects have been the identification of a manufacturing partner in hyposcasi and the partnership with Sarepta, another biotech in the States with a lot of expertise in taking medicinal product forward for Duchenne muscular dystrophy. The gene therapy specific microdystrophin developed by Geneton has dissimilarities with existing gene therapy product, although broadly speaking they are a not completely different family. Specific differences are related to the fact that the promoter that regulates the expression of the strand gene is different. The sequence of the microdystrophin is also different. And studies that have been done using this microdystrophin in animal model for muscular dystrophy have indicated a good safety profile, but also efficacy at low doses that is encouraging. The study 104 is the pre-inclusion on natural history study. It will occur, and is started to recruit in France, but will occur in 4 countries, in the States, in the UK, in France, and in Israel. We will recruit up to 100 individuals with Duchenne who are still ambulant between the age of 5 and 9 years old. And the study will last a minimum of 6 months, up to 3 years, and will be a prerequisite for recruiting children into the subsequent AAV gene therapy study. Regarding the specific medicinal product, the one important difference compared to other existing AAV gene therapy currently in clinical trial is the serotype. This will be a serotype 8. In 2015, Genethon has already received the advice from EMA, the European Medicine Agency, regarding the protocol and in the protocol assistance. In 2020, there has been the submission to the French authorities, and the submission in UK is ongoing. And more recently, the ethics committee in France have approved the natural history study, and the first patient have already been recruited in France. The clinical trial will recruit in the same centers and in the same patient who have been recruited in the original natural history study. So in conclusion, this new AAV8 vector containing this new microdystrophin gene appears to be a promising treatment for Duchenne muscular dystrophy based on the preclinical animal models. A natural history study, the GNT-014, is already started in some countries and will now hopefully start to recruit in the remaining country, and will be necessary to identify patient eligible for the future gene therapy study. And the gene therapy protocol has been approved by the French Ethics Committee, and there is pending for the competent authority decision. And if everything goes well, the first in-human trial is planned for the first part of 2021. Thank you very much, and good evening.
I would like to thank the companies, the presenters for the presentation. Thank you so much. And we will now move to the Q&A session, which is a live session. And for this, we have joining us live on Zoom today, virtually, Dr. Singh from Sarepta, Professor Nevo on behalf of Pfizer from Schneider, Dr. Schneider from Solid-Bio, and Professor Francesco Montuni on behalf of Genethon. So thank you all for joining us this evening, and we will start with the questions. So the first question will be for Professor Nevo. Many regarding the Pfizer study. So many families have been waiting patiently for the opening of the first clinical trial for gene therapy here in Israel. When is the study supposed to start?
Well, we have this week in Snyder's as well as in Hadassah the initiation of the study. With also Helsinki IRB confirmation already. So we assume that the study will start in the next, let's say, couple of months.
Okay, thank you. Thanks a lot. Thank you very much. We will now move to the second question, and this is for Professor Montoni. This is a delicate question. If a child does develop antibodies against the AAV during the first year, will this prevent him from getting the treatment eventually? Or will he have something else to offer him? Could he be able to continue to participate in the trial?
is an important question, as you say, is a delicate question and requires an honest answer. I think as things stand today, it will not be possible to recruit a child who has developed an AAV positivity into an AAV gene therapy trial. The same, of course, applies for people not just recruited, is not to be recruited in the natural history study, increase the probability to develop antibodies. And in children, the frequency of antibodies against most of these AAV is pretty low. However, in all honesty, if a child was either coming from the natural history study or just recruited fresh, and he had high titer of antibodies against one of these AAV, things stand today, no, that child cannot be dosed with AAV. There is work to try to see how to overcome this in the future, but this is where we are today.
So another question for you, and maybe also for the others. If a child is recruited to the placebo group, and he is recruited and he gets the placebo, and then during the first year he becomes seropositive, and he has antibodies, what will happen? He will just go out from the study? Does the company have something else to offer him? I start with you, but I would like to maybe to get answer from Professor ניוון, and from Dr. Schneider, from Dr. Soon. What happens if that? Do you think it's a possibility, a realistic possibility, that a child will become positive after he gets the placebo?
Well, I will start, and then my colleagues can follow. I mean, in a way, it's not very different from that of most clinical trials when we recruit children into a placebo clinical trial, and the child, for some reason, becomes not eligible to receive the active treatment. This makes that child, so if that was an antisense trial, this was a pharmacological agent, and then the individual develops for example, a liver involvement as an example, and cannot be recruited into the active treatment, the child will be excluded from that follow-on potential therapeutic intervention. So to some extent, this is not unique of AAV gene therapy trials.
Can follow up.
Yes.
No, as Dr. Montoni said, this is first of all, patient participation in these trials is incredibly important. And understanding that there are, of course, always risks in enrolling either in a treatment cohort or in a placebo cohort. At the end of the day, I think while currently turning positive, so converting to seropositive antibody levels, would preclude those children from being dosed immediately, there are a lot of promising research avenues underway to identify in the future how we can potentially dose those patients. Many different approaches using antibody-based approaches, small molecules are all being explored so that in the future, pre-existing antibodies are not a limitation of patient enrollment in these gene therapy trials. And while that research is still very early, I think it's very encouraging that this has been identified as an issue that we have to overcome and tackle so that all patients can receive a gene therapy approach regardless of whether or not they have pre-existing antibodies at the onset.
Okay. I have a question for Professor ניוון. If a child gets now recruited to the gene therapy study with AAV9, will it prohibit him to be involved in the future in different studies or to get a therapy with AAV8, for example, from a different company? It's two different questions, but it's kind of, yes, it touches the same question.
I would like to start and just and continue what the previous speaker said, is that there are risks in being in clinical studies. However, most studies, the placebo group is smaller than the treated group. In the Pfizer study, it's 2-to-1. In the very preliminary study, as I showed in my presentation, there was something only of 10% who initially had antibodies. So there is always a chance of getting the antibodies within the year between the placebo and the treatment schedule, but it's not a very it's not very common that we expect it to happen. So this is one thing. In terms of the questions, I'm not sure there's a lot of data, but I assume that if you have antibodies to AAV9, you probably won't be able to get the AAV8, but I would be happy to hear my other colleagues about this issue.
Yes, Dr. Singh, you want to add something regarding the options of switching from one gene therapy to another capsid, to another virus? Is it a possibility?
Great question. Short answer, in the clinical trials, you'd be excluded because we don't want to confound the results of the trial with the previous therapy. In the commercial setting, once if the drug is found to be effective and safe and has benefit, then really you'll have to actually follow the label for the commercial drug, which means screening for the antibody specific to that agent. Now, there is some overlap between AAV8, RH74, AAV9, and AAV8, but it's not 100%. So there is a possibility that, if you received one, you may be antibody positive for the one you received, but still be negative for a future therapy. But I think we'd have to look at that when the data comes out from the trials. And then the patients will need to be screened for the particular requirements for the drug that they're intended to potentially receive.
Okay. Thank you, Dr. Singh. I want to ask you some more questions regarding the presentation. We learned about this bridging study, the 103. Is it going to be in parallel with your phase 3 international clinical study, or will the phase 3 international study will be postponed until you have the results of the 103 bridging study?
No, wonderful question. So we've actually already started the 103 study. So that started in the United States, and our first patient's being dosed in that trial. It's not linked in terms of gating to the 301, which is our phase 3 study. For a phase 3 study, we need to have some other data with regards to the attributes of the drug, and we'll be submitting that when that's available. But they're not gating. We're not delaying the start of the phase 3 study based on the 103 full results. Now, the 103 results that we'll generate beforehand will help support our submissions for that phase 3 study.
Thank you very much. It's very great to hear. I wanted to ask you something else. In the lecture, it was mentioned that Sarepta-Roche is planning a clinical trial for non-ambulatory patients. Could you please elaborate on that regarding timelines, or when do you think it will be a possibility for the community?
Thank you again. As Alex pointed out, yes, we are planning the non-ambulatory study. That's study 303. We will have sites in Israel for that study, and I defer to some of the colleagues on the call that actually know a little bit of the advice they've given us on how to conduct it. The timing is dependent on our interactions with agencies on actually when we could start initiating it, but our plans is to have that up and running towards the later part of next year, so the fall of 2021.
Oh, that is great news. Professor Navarro, can you please elaborate a little bit more about the reason for the serious adverse events in the Pfizer study? Was it because of the capsid, of the specific choice of AAV9, or was it because of the micro-dystrophin, the gene particles? Do you have any on the serious? And what did you do? What measures did Pfizer do to prevent such effects in the next study?
We don't have too much inside information from Pfizer. So, but what I know is there will be increased prednisone preventive treatment dose, which hopefully will decrease the chance of having these side effects. We are prepared for the eculizumab treatment, and all of the patients in Israel will be vaccinated against meningococc, and actually different from what happens either in Europe or in the States. We'll have two actually vaccines which will actually address the strains that are prevalent in Israel. We don't have too much information exactly what component or what's the reason for the specific side effects that happened in the phase 1 of the trial.
Thank you. The next question is for both Dr. Joel Schneider and Dr. Singh. Pfizer reports about 10% of participants at screening having an antibody against the AAV virus. Did you observe the similar numbers in your screening process? Dr. Schneider?
Sure. You know, we haven't gone into a lot of detail about what we've seen inside of the actual trial. As you're aware, we've dosed 6 patients to date, and we're going to get a larger data set together, really identify within our own sample set how many patients are positive for AAV9, and then in the future share some of that information.
Okay, Dr. Singh, can you elaborate on that regarding the percentage of the AAV positive? Was it also around 10%, like the Pfizer disclosed?
Yes, for R74, we looked at 94 patients. We screened them that both had DMD and limb girdle, since the platform is the same, and the percentage pre-existing antibodies is about 15%, slightly less than DMD. And we're actually doing another study starting later this month, a seroprevalence study with 300 subjects that we'll be screening to see what the true incidence of prevalence is pre-existing.
I have another question for you. Can you please tell us when you think we will know about the phase 2 study, the clinical study that Sarepta did, recruiting 42 patients that were given the gene therapy?
Sure. So we're just finishing up that first part, and it was 41 subjects. We expect to have the last portion of the data in later this month, and hope to share results of that first portion early part of next year, January, February of next year.
That's wonderful news. So with that happy note, I want to thank everyone for your time and sharing us the information and participating and coming virtually to Israel. And we hope to see you in person as soon as it will be possible. So thank you very much, everyone.
Thank you for your support.
Thank you.
תודה רבה, דוקטור דור. תודה רבה לכל המרצים במושב ולמשתתפים בפאנל. אני רוצה לספר לכם שבדיוק היום אנחנו יוצאים לדרך עם קמפיין ה-500. מה זה אומר? בעזרתכם אנחנו רוצים להגיע תוך שבועיים ל-500 מעגלים לטובה עבור עמותת צעדים קטנים. מה מעגלים? מעגלים בצורה קבועה מכל רכישה בכרטיס האשראי את ההפרש לשקל הקרוב. לדוגמה, חיוב על רכישה בסופר בסכום של 78 שקלים ו-20 אגורות יעוגל לסכום של 79 שקלים. כלומר, תרמתם רק 80 אגורות. בשבילכם זה ממש כסף קטן, נשמע ממש מעט, אבל מדובר בשקלים בודדים בחודש בשבילכם. אבל יחד עם כל המעגלים זה יוצר סכום גדול ומשמעותי שיסייע לנו לקדם פרויקט נוסף חשוב בשביל המשפחות שלנו. אז חסרים לנו רק עוד 125 מעגלים כדי להגיע ליעד. תיכנסו לצ'אט, יש שם קישור, תצטרפו עכשיו, תשתפו עוד חברים, ו3 מתוך המצטרפים בתקופת הקמפיין יזכו בשובר של 500 שקלים לרכישה במגוון בתי עסק ומסעדות. תודה רבה שהייתם איתנו כאן הערב, ניפגש שוב בעוד כחצי שעה, בשעה 8:30 בדיוק. תודה רבה.
השנה נחגוג יום הולדת 10 לעמותה. מוזמנים להגיע לטובה לצעדים קטנים. עיגול קטן שלכם שווה חיבוק גדול עבורם. מה זה עיגול לטובה אתם שואלים? זה רעיון פשוט גאוני שמאפשר לתרום את עודף האגורות בכרטיס האשראי לעמותה שאתם בוחרים. כמו צעדים קטנים. כמה שקלים בכל חודש שבזכות מאות אלפי מעגלים לטובה משנים חיים כל יום. אם היה לי כרטיס אשראי, גם אני הייתי מעגל, בחיי.